Hi, I'm Nathan Hillson, and this is the first part of a series of JBEI tutorials for its ICE software. ICE is the platform that JBEI uses to store and manage all of its DNA sequences, microbial strains, DNA parts, and Arabidopsis seeds. In this series, we're going to be going through a variety of different topics. In this first part, we're going to be just covering the basics of how you browse and search through entries. In the second part, we're going to be looking at Vector Editor and how you can edit and annotate DNA sequences. Then we're going to be looking in the third part into some more types of information that might be associated with some of these entries. In the fourth part, we'll show you how to create your own entries. In the fifth part, we'll talk about how you can share those entries with others. And in the last part, part 6, we'll be going after how you can actually get strains out of the repository and how you can export data. So without further ado, let's go ahead and go and see how we can browse and search through ICE. In this first demo of the ICE registry platform, we're going to be going over some very basic types of things. So the first thing that we're going to do is just go ahead and go to the public instance of the JBEI ICE platform, public-registry.jbei.org. If you don't already have an account, all you have to do is click on the Create Account button, fill in your details, submit, you'll be emailed a temporary password, and you can use that to log in. So I already have an account, so I'm just going to go ahead and log in. And once you log in, this is kind of basically going to be what you're going to see. So by default, it's just going to kind of show all of your personal entries. So these would be all of our entries. So the kind of the type of the entry, so it could be a part or a plasmid or a strain or a seed. It would have kind of like an ID, a name, and kind of like a summary description, and some other types of information, like what's its status, does it have any physical samples, does it have a DNA sequence associated with it, when it was created, when it's, how often it's been viewed, etc. You can also kind of just like cover over one of the IDs to get a little bit more information. For example, who is the principal investigator, or when it was last modified, biosafety level, and so on. So if you click on the part ID, now you can kind of get into that more detail. And you also can kind of see a representation of the sequence. Now, if you kind of want to just kind of page around between different entries, you can kind of just click on these paging icons and kind of page through your various entries. Now, in addition to kind of your personal sets of entries, there's also kind of a featured set of collections. Now, in the JBEI public registry, we have a collection for each of our publications. So say, for example, for this particular paper, these are going to be all of the plasmids and strains and parts that were associated with that particular paper. So that's basically kind of how you browse through entries, browse through collections. Some things to kind of note, if you kind of scroll down on the left-hand panel side there, you'll also kind of see this Web of Registries section. So you can actually browse through other registries besides the public JBEI registry. So for example, if I want to look at the Joint Genome Institute registry, I can click on that and I can see all of its entries. In this case, these would be kind of the DNA vectors that it uses in its synthetic DNA program. So in addition to kind of just browsing through the entries in ICE, you can also search. So you can search for things like maybe the name of a particular vector that you're interested in. So maybe I'll search for this PBB A0K-RFP. And one kind of pro tip that you can do when you're searching, when ICE is searching for things, it'll try to come up with things that not only have the identical hit, but also might have things that are pretty similar. So if you know exactly what you're looking for, you can put the search term in quotes, and then basically it'll really rank higher those things that have that exact potential match. So then you can find these things you're looking for. Now, interestingly, in addition to just the local search results, you can also search other registries. So in addition to just browsing these other registries like the JBEI registry or the SIMBRIC registry, you can also kind of search for those. And in this particular case, I also find this particular vector in the SIMBRIC registry, and I can browse it just as if it were actually within the JBEI public registry. So those are some of the basics over, kind of creating accounts if you need one, how to kind of browse entries, browse collections, browse through the web registries, and do some searching. Maybe one last thing to show you is that in addition to kind of do a standard type of a search, you can also do what we would call an advanced search. So you can search for things like, for example, maybe what are all the entries that have a biosafety level 2, or you can blast for particular sequences. So if I do those types of things, now in the local registry, I can see all of the entries that are BL level 2, or I can look in the other registries and see in the Symbrick registry there's also a couple of BL2 level entries. All right, so that's how you basically browse and search through ICE. In the next part 2 coming up, we're going to go into Vector Editor, and you can see how you can actually edit and annotate DNA sequences.